The world around us hums with life, each creature carrying secrets written into its very cells, strategies refined across millions of years. Tonight, we'll discover how nature has solved some of its most impossible challenges. Welcome to Bedtime Biology, where science meets serenity. Each night, we explore the quiet wonders of the natural world, calming your mind and sparking your curiosity. Please like, subscribe, and comment below with where you're checking in from. From the frozen wastes of Antarctica to the crushing depths of the ocean floor, from scorching deserts to the perpetual darkness of deep-sea trenches, life has found a way, not just to survive, but to thrive. The strategies that evolution has crafted are more ingenious than any human invention, more elegant than any machine we could design. Some animals can pause their lives entirely, entering states of suspended animation that would be science fiction if we hadn't witnessed them with our own eyes. Others have mastered the art of invisibility, blending so seamlessly with their surroundings that they seem to vanish into thin air. Some have formed partnerships so intimate that neither partner can survive without the other. And still others possess abilities that seem to defy the very laws of biology, regrowing entire limbs, organs, even portions of their brains, as casually as we might grow a new fingernail. Tonight, we're going to slow down and truly see these wonders. We'll watch as a tiny water bear, no bigger than a speck of dust, survives conditions that would destroy any human-made spacecraft. We'll witness the chemical magic that allows creatures to produce their own light in the eternal night of the deep ocean. We'll follow animals on journeys that span entire continents, guided by invisible forces that we're only beginning to understand. And as we explore each strategy, each adaptation, we'll see not just the mechanics of survival, but the profound interconnectedness of all life on Earth. Every creature we encounter tonight has something to teach us, something that might one day help us solve our own challenges, heal our own bodies, or simply see the world in a new way. So settle in, breathe deeply, and prepare to discover that reality is far stranger, far more beautiful, and far more intricate than any story we could imagine. The masters of suspended animation, in the quiet spaces between the drops of morning dew, in the film of water coating moss on a stone wall, in the thin layer of moisture on a leaf, there lives a creature that has mastered something humans have long dreamed of: the ability to pause life itself. This creature is the tardigrade, and though you may never have seen one, it may well be one of the most remarkable animals on Earth. Tardigrades, sometimes called water bears or moss piglets, are microscopic animals, typically measuring between 300 and 500 micrometers in length. That's about half the width of a human hair. Under a microscope, they appear both alien and oddly charming, with plump, segmented bodies and 4 pairs of stubby legs ending in tiny claws. They lumber through their watery microworlds with an endearing, bear-like gait that inspired their common name. But it's what these tiny creatures can do, not what they look like, that makes them extraordinary. When a tardigrade's environment begins to dry out, something remarkable happens. The animal doesn't simply die or try to flee. Instead, it enters a state called anhydrobiosis, from the Greek words meaning life without water. Over the course of several hours, the tardigrade begins to lose water from its body, transforming as the moisture leaves its cells. It pulls its legs inward, retracts its head, and curls into a tight barrel shape called a tun. In this state, it loses more than 95% of its body water. For most organisms, this would be catastrophic. Water is essential for virtually every biological process. It's the medium in which chemical reactions occur, the solvent that allows molecules to move and interact, the structure that gives cells their shape. Remove water and proteins denature, cell membranes collapse, DNA breaks apart. Death follows quickly, but the tardigrade doesn't die. Instead, it enters a form of cryptobiosis, a state of life so reduced that it barely qualifies as life at all. Cryptobiosis, meaning hidden life, is a broader term encompassing several types of suspended animation. Anhydrobiosis is just one form. There's also cryobiosis, induced by freezing, anoxybiosis, triggered by lack of oxygen, osmobiosis, caused by high concentrations of dissolved substances, and possibly chemobiosis, brought on by toxic chemicals. In each case, the animal's metabolism doesn't just slow down, it essentially stops. Scientists have measured the metabolic rate of tardigrades in cryptobiosis at less than 1/100% of their normal rate. For all practical purposes, they're in stasis. The question that has fascinated scientists for decades is how? How does a tardigrade survive what should be unsurvivable? The answer, we've learned, is extraordinarily complex, involving multiple molecular strategies working in concert. One key player is a sugar called trehalose, a disaccharide made of two glucose molecules. As water leaves the tardigrade's cells, trehalose accumulates, replacing the water molecules. In doing so, it helps stabilize proteins and cell membranes, preventing them from collapsing or crystallizing. It's like packing fragile glassware in foam peanuts. The trehalose molecules fill the spaces, cushioning and protecting the delicate cellular machinery. But trehalose is just part of the story. Recent research has uncovered another crucial element: proteins that are unique to tardigrades. These proteins, called Cars proteins, after the word cytoplasmic abundant heat soluble, form gel-like structures inside cells as they dry out. These gels appear to physically stabilize cellular components, preventing them from moving around and becoming damaged. When water returns and the tardigrade rehydrates, these gels dissolve, releasing the cellular machinery unharmed. It's elegant and beautifully simple, a molecular cage that protects and releases. Tardigrades also face another challenge during anhydrobiosis: the accumulation of damaged molecules and particularly damaged DNA. In a normal, active state, cells constantly repair small amounts of DNA damage caused by cosmic radiation, chemical reactions, and simple copying errors. But in cryptobiosis, with metabolism at a standstill, this repair work can't happen. Damage accumulates. When the tardigrade rehydrates, it faces a backlog of repairs that must be completed before normal life can resume. Research has shown that tardigrades possess enhanced DNA repair mechanisms that spring into action during rehydration. They also produce elevated levels of antioxidants, molecules that neutralize harmful reactive oxygen species that form when metabolism restarts. It's as if the tardigrade, upon waking, runs a complete diagnostic and repair program before fully coming online. The capabilities of tardigrades in cryptobiosis are almost unbelievable. Scientists have exposed desiccated tardigrades to temperatures as low as -272 degrees Celsius, just 1 degree above absolute zero, the coldest temperature theoretically possible. The tardigrades survived. They've been heated to 150 degrees Celsius. They survived. They've been subjected to pressures 6 times greater than those found in the deepest ocean trenches. They survived. They've been blasted with radiation doses over 1,000 times higher than would kill a human. They survived. In 2007, tardigrades were even sent into space, exposed to the vacuum and solar radiation for 10 days. When brought back to Earth and rehydrated, many resumed normal activity, and some even reproduced successfully. But tardigrades aren't the only animals that have mastered cryptobiosis. In the northern forests of North America, another unlikely survivor emerges each spring from beneath the snow and frozen soil. This is the wood frog, Rana sylvatica, an amphibian that looks utterly ordinary, with moist brown or tan skin, a dark eye mask, and a body barely 3 inches long. Yet this small frog does something that seems impossible for a vertebrate animal. It freezes solid and survives. As autumn gives way to winter and temperatures drop, the wood frog seeks shelter beneath leaf litter or logs. It doesn't burrow deep underground like other hibernating animals. Instead, it remains relatively close to the surface, where temperatures can plunge well below freezing. As the cold intensifies, ice begins to form on the frog's skin. This ice formation triggers a cascade of responses. Special proteins in the frog's tissues detect the presence of ice crystals and immediately begin mobilizing glucose from the liver. This glucose floods into the bloodstream and then into the cells, reaching concentrations 50 to 100 times higher than normal. Why glucose? Like trehalose in tardigrades, glucose acts as a cryoprotectant, a molecule that lowers the freezing point of cellular fluids and stabilizes cell membranes and proteins. As ice continues to form in the spaces between cells and in body cavities, the concentrated glucose inside cells prevents them from freezing solid. It's a delicate balance. Ice forms in up to 65% of the frog's body water, but the cells themselves, protected by their glucose shields, remain unfrozen. Meanwhile, the frog's heart stops beating, its blood stops flowing, its brain ceases all electrical activity, its lungs stop exchanging gases. By every conventional measure, the frog appears dead. It's stiff, frozen, unresponsive. If you were to pick it up, it would be as solid as a small ice cube. Yet inside, in a state of profound hypothermia, the cells remain alive, their internal machinery preserved by glucose and other cryoprotectants, including urea, another small molecule that provides additional protection. The wood frog can remain in this state for weeks or even months throughout the harshest part of winter. Then, as spring arrives and temperatures rise, something extraordinary happens. The ice begins to melt, starting from the frog's core and radiating outward. As water returns to liquid form, the heart, which has been frozen solid for months, begins to beat again. Slowly at first, an irregular flutter, then gradually strengthening into a steady rhythm. Blood begins to flow. The brain restarts. Within a day or two of thawing, the frog is hopping around, ready to mate, feed, and live as if nothing unusual had happened. The fact that a vertebrate can survive freezing challenges many of our assumptions about biology. For decades, scientists thought that ice formation in tissues was invariably fatal to complex animals. Ice crystals, sharp and growing, puncture cell membranes. Freezing concentrates dissolved substances, creating toxic conditions. The absence of circulation means no oxygen delivery, no waste removal. How could any animal, let alone one with a brain, a heart, and complex organs, survive this? The answer lies not just in the cryoprotectants, but in the controlled nature of the freezing. Wood frogs don't prevent freezing, they manage it. Special ice-nucleating proteins in their tissues actually promote ice formation in specific locations, particularly in body cavities and between cells. This might seem counterintuitive, but it serves a crucial purpose. By controlling where ice forms, the frog ensures that it forms slowly and safely, rather than rapidly and catastrophically. The ice crystals that form are small and rounded, rather than large and jagged. This controlled freezing prevents the kind of damage that uncontrolled freezing would cause. Moreover, recent research has shown that wood frogs possess other adaptations that help them survive freezing. Their tissues are more resistant to ischemia, the lack of oxygen and nutrients that occurs when blood flow stops. They have enhanced mechanisms for dealing with the oxidative stress that occurs when metabolism restarts. Their cells are better at managing changes in ion concentrations that occur during freezing and thawing. It's a comprehensive survival system, not a single trick, but an orchestra of adaptations working together. Wood frogs aren't alone in their freeze tolerance. Several other frog species, particularly those living in cold climates, can survive freezing, though often not as extensively as wood frogs. Some turtles, living in similar environments, can also survive partial freezing. Certain insects, including some species of beetles and flies, routinely freeze solid in winter and revive in the spring. Each of these animals has evolved similar yet distinct strategies, convergent solutions to the same fundamental problem: how to survive when the water in your body turns to ice. The mechanisms of cryptobiosis and freeze tolerance hold profound implications beyond simple biological curiosity. Medical researchers are intensely interested in these phenomena because they might offer solutions to some of medicine's most challenging problems. Organ transplantation, for example, is limited by the fact that organs rapidly deteriorate once removed from the body. Current preservation methods can keep organs viable for only hours, or, at most, days. If we could understand and replicate the molecular mechanisms that allow tardigrades to survive desiccation or wood frogs to survive freezing, we might be able to preserve organs for weeks, months, or even longer. Patients wouldn't die waiting for matching organs. Surgeries could be scheduled electively rather than as emergencies. The entire infrastructure of transplantation could be transformed. Similarly, the study of cryptobiosis has applications in preserving vaccines, blood products, and other biological materials. Many vaccines require constant refrigeration, making them difficult to distribute in areas without reliable electricity. If vaccines could be dried and stabilized using principles derived from tardigrade biology, they could be shipped and stored at room temperature, dramatically expanding access to life-saving immunizations. Some researchers are already working on dry vaccines based on these principles. The lessons from these masters of suspended animation extend even further into questions about the nature of life itself. If an organism can have essentially no metabolism for years or even decades, and then resume normal function, what does it mean to be alive? Is a tardigrade in anhydrobiosis alive? It has no measurable metabolism, no response to stimuli, no apparent awareness. Yet, it's not dead, because it can return to full function. It exists in a liminal state between life and death that challenges our categories and definitions. In the end, these creatures remind us that life is more resilient, more adaptable, and more creative than we often imagine. the mechanisms that allow a water bear to survive the vacuum of space, or a frog to freeze solid and revive, aren't just curiosities. They're demonstrations of the profound elegance of evolution. Each adaptation is the product of millions of years of refinement, of countless generations testing small variations, of nature solving problems in ways that human engineers are only beginning to appreciate. When we look at a tardigrade under a microscope, we're not just seeing a small animal. We're seeing a masterpiece of molecular engineering, a testament to the power of natural selection to craft solutions to impossible problems. And in that tiny, transparent body, barely visible to the naked eye, lies knowledge that might one day help us preserve organs, cure diseases, or even, perhaps, make the long journeys to other worlds that tardigrades have already survived. The art of disguise and deception. If you've ever walked through a forest and suddenly realized that what you thought was a stick is actually an insect, or noticed that what appeared to be a dead leaf is in fact very much alive. You've witnessed one of nature's most ancient and effective survival strategies, camouflage. The ability to avoid being seen, to blend seamlessly with one's surroundings, or to mimic something unpalatable or dangerous can mean the difference between life and death. For prey, it's a way to avoid becoming a meal. For predators, it's a means of approaching victims undetected. And throughout the animal kingdom, the arts of disguise and deception have been refined to such extraordinary levels that the line between organism and environment can become almost impossible to discern. The most straightforward form of camouflage is called background matching, where an animal's coloration, pattern, and sometimes even texture closely resembles its typical habitat. This strategy is ubiquitous, seen in everything from the tawny fur of a lion that blends with dry grasslands to the speckled pattern of a flounder that mirrors the sandy seafloor. The effectiveness of background matching depends on the visual system of the animal's predators or prey. Because most predators rely heavily on vision to detect movement and recognize shapes, anything that breaks up the recognizable outline of prey makes detection much more difficult. Consider the ptarmigan, a bird that lives in Arctic and alpine environments. In summer, the ptarmigan's plumage is mottled brown and grey, matching the rocks, lichens, and low vegetation of its habitat. But as winter approaches and snow begins to cover the landscape, the ptarmigan molts, gradually replacing its summer feathers with pure white ones. By the time deep winter arrives, the bird is completely white, virtually invisible against the snow. This seasonal colour change, seen also in Arctic foxes, snowshoe hares, and stoats, is triggered by changes in day length that affect hormone production, which in turn controls feather or fur pigmentation. It's a remarkable example of how animals can actively maintain their camouflage as their environment changes. But background matching is just the beginning. Another camouflage strategy is called countershading, a pattern in which an animal is darker on its upper side and lighter on its belly. At first glance, this might seem counterintuitive. Wouldn't being the same color all over provide better concealment? The answer is no, and the reason has to do with how light and shadow work. When light comes from above, as it normally does, it illuminates the top surfaces of an object while leaving the bottom surfaces in shadow. This creates a gradient that makes the object appear three-dimensional. A uniformly colored sphere, for example, will look darker on its underside, even if it's painted a single color. Countershading counteracts this effect By being darker on top and lighter below, an animal can cancel out the natural shading pattern, making its body appear flat and uniform rather than three-dimensional. Sharks are a classic example. Most sharks have dark backs and white bellies. When viewed from above, against the dark depths, the shark's dark back blends in. When viewed from below, against the lighter surface water, the pale belly is less visible, but the counter-shading also helps break up the shark's outline, making it harder for both prey and predators to recognize its shape. Yet, even more impressive are the animals that don't just match colors, but can change them at will. Cephalopods, the group that includes octopuses, squids, and cuttlefish, are the undisputed masters of this art. The skin of a cephalopod is one of the most sophisticated displays in nature, capable of changing color, pattern, and even texture in less than a second. This is accomplished through specialized cells in the skin called chromatophores. Each chromatophore is like a tiny elastic sac filled with pigment. Attached to the sac are radial muscles that can expand or contract it. When the muscles relax, the chromatophore shrinks to a tiny dot, and its color is barely visible. When the muscles contract, pulling the sac outward, the pigment spreads over a much larger area, and the color becomes vivid. A cuttlefish's skin contains thousands of these chromatophores, arranged in layers with different pigment colors. The top layer typically contains yellow or red pigments. Below that are chromatophores with darker browns and oranges. Beneath all the chromatophores are other specialized cells, iridophores, which contain stacks of reflecting plates that can produce iridescent greens, blues, and silvers, and leucophores, which scatter light to produce white coloration. By independently controlling each of these cells, the cuttlefish can produce an almost infinite variety of colors and patterns. What makes this even more remarkable is the speed and precision with which these changes occur. A cuttlefish can assess its background and adjust its appearance in a fraction of a second. Researchers have shown that cuttlefish can match not just the general color of a surface, but its texture. If placed on a checkerboard pattern, the cuttlefish will produce a checkerboard pattern on its skin. If the squares are too small for the cuttlefish's resolution limit, it will produce a mottled grey, effectively averaging out the pattern. This dynamic camouflage works so well that a cuttlefish settling onto a complex substrate seems to dissolve, its outline becoming impossible to trace. But the ability of cephalopods to change their appearance isn't only about matching backgrounds. They also use color changes for communication, displaying vivid patterns during courtship or territorial disputes. Male cuttlefish, competing for females, will flash waves of color across their bodies, each pattern conveying information about size, fitness, and aggressive intent. In some species, males have even evolved a sneaky strategy. When a large, dominant male is present, a smaller male will disguise himself by retracting his male display arms and adopting female coloration. This allows him to approach females without triggering an attack from the dominant male, essentially cross-dressing to bypass competition. The sophistication of cephalopod camouflage becomes even more intriguing when you consider that most cephalopods are colorblind. Their eyes contain only one type of light-sensitive pigment, meaning they see the world in shades of grey. How then can they so accurately match colors they cannot see? Recent research suggests that cephalopods may use their skin itself as a light sensor. Chromatophores and the surrounding cells may contain light-sensitive proteins similar to those found in the eyes, allowing the skin to directly sense and respond to light without input from the brain. This would be like having millions of tiny eyes spread across the body, each one sensing local lighting conditions and adjusting accordingly. It's a hypothesis still being tested, but if true, it would represent a completely novel form of sensing and response. Beyond simple color matching, many animals employ a strategy called disruptive coloration. Rather than blending with the background, disruptive coloration works by breaking up the recognizable outline of the animal's body, making it hard to see where the animal begins and ends. Zebras are a famous example. Their bold black and white stripes make individual animals stand out. But when zebras gather in herds, the overlapping stripes create a confusing visual pattern that makes it difficult for predators like lions to single out and target one individual. Studies have shown that lions have lower hunting success rates when attacking zebra herds compared to hunting other similarly sized prey. The stripes also serve another function. They may confuse biting insects. Experiments with striped patterns painted on horses show that horseflies approach and land on striped surfaces far less frequently than on solid colored ones, possibly because the stripes create visual interference patterns that the insects cannot navigate. Some of the most extreme forms of camouflage involve not just matching the environment, but actually mimicking specific objects within it. This is called masquerade, and it reaches its pinnacle in insects like walking sticks and leaf insects. A walking stick looks so much like a twig that it's almost impossible to identify as an animal unless it moves. The body is long, thin, and cylindrical, often with small bumps and irregularities that resemble bark texture. The legs are held at angles that mimic the position of small branches. Coloration matches dead wood, sometimes with hints of green or brown to suggest lichen. When disturbed, many walking stick species will sway gently from side to side, mimicking a twig moving in the wind, a behavior that reinforces the illusion. Leaf insects take masquerade even further. Their flattened bodies are shaped exactly like leaves, complete with vein-like patterns running across their wings and legs that look like leaf stems. Some species have brown patches on their green leaves that look like dead or damaged tissue, or edges that appear chewed by caterpillars. When resting on a branch, a leaf insect will often position its legs to merge with its body outline, creating a more leaf-like shape. And like walking sticks, they may sway when there's a breeze, completing the illusion. A predator scanning for prey sees twigs and leaves, not potential food. Camouflage is one thing, but mimicry is another. In mimicry, one species evolves to resemble another species, gaining protection or advantage from the resemblance. The most common form is called Batesian mimicry, named after the 19th century naturalist Henry Walter Bates. In Batesian mimicry, a harmless species evolves to look like a harmful or unpalatable one. Predators that have learned to avoid the dangerous model will also avoid the harmless mimic, even though the mimic poses no actual threat. A classic example is the resemblance between harmless hoverflies and stinging bees or wasps. Hoverflies are true flies, with no stingers, no venom, completely defenseless. But many species have evolved yellow and black striped patterns that make them look remarkably similar to bees or wasps. Birds that have been stung by a wasp will be wary of anything with yellow and black stripes, including hoverflies. The hoverfly benefits from this learned avoidance without having to invest in the metabolic cost of producing venom or growing a stinger. It's dishonest signaling, but it works. For Batesian mimicry to be effective, the model must be more common than the mimic. If mimics become too numerous, predators will encounter them more often than they encounter the genuine dangerous species, and they'll learn that the pattern doesn't always mean danger. The protection breaks down. This limits the success and abundance of mimics, creating an evolutionary balance. Another form of mimicry is Müllerian mimicry, named after the German naturalist Fritz Müller. In Müllerian mimicry, multiple unpalatable or dangerous species evolved to resemble each other. This might seem paradoxical at first. Why would two already protected species benefit from looking alike? But the advantage is clear. It speeds up learning for predators. If a predator encounters a poisonous frog with red and black stripes, it learns to avoid red and black frogs. If multiple species of poisonous frogs all share the same red and black pattern, the predator learns faster and remembers longer. The evolutionary cost of teaching predators to avoid you is shared among all the similar-looking species, reducing the number of individuals that must be eaten for predators to learn the lesson. Perhaps the most unsettling form of mimicry is aggressive mimicry, where a predator mimics something attractive or harmless to lure prey. The anglerfish does this literally, with a modified dorsal spine that hangs in front of its mouth like a fishing lure, often tipped with a glowing bacterial culture. Smaller fish, attracted to what appears to be a morsel of food or a glowing plankton, swim closer to investigate, and are suddenly engulfed by the anglerfish's enormous jaws. Fireflies of the genus Futurus use aggressive mimicry by imitating the flash patterns of females from other firefly species. When males of the other species approach, expecting to mate, the Futurus female eats them. In all these forms of camouflage, masquerade, and mimicry, we see evolution as a creative force, sculpting bodies, colors, and behaviors to solve the fundamental problem of survival. Each strategy comes with costs and benefits, trade-offs that evolution has navigated over millions of years. Camouflage requires maintaining specific coloration, which may be energetically expensive, and may limit where an animal can live. Mimicry depends on the presence and behavior of other species. Disruptive coloration might hinder species recognition, making it harder for individuals to find mates. Yet, despite these constraints, the diversity and sophistication of these strategies are breathtaking. They remind us that evolution is not random, but powerfully shaped by the environments organisms inhabit and the other species with which they interact. Every moth that looks like bark, every butterfly that resembles a leaf, every octopus that vanishes against coral is a solution to a specific problem, tested and refined through countless generations. And as we study these solutions, we find applications for human technology, military camouflage, adaptive fabrics, even computer vision systems that must distinguish objects from backgrounds. The animals were solving these problems long before we arrived, and they remain the masters of the art. Living together, partnerships that save lives in the intricate web of life. No organism exists in isolation. Every animal, plant, fungus, and microbe is surrounded by others, and the relationships that form between species can be as important to survival as any physical adaptation. Among the most fascinating of these relationships are mutualisms, partnerships where both participants benefit. These alliances shape ecosystems, enable life in otherwise impossible environments, and demonstrate that cooperation, not just competition, is a fundamental force in evolution. Perhaps the most visible example of mutualism is the relationship between flowering plants and their pollinators. A bee visiting a flower is not just randomly moving through the environment, it's participating in an ancient partnership refined over millions of years. The flower offers nectar, a sugar-rich liquid that provides energy, and pollen, which contains protein and fats. The bee, in return, carries pollen from one flower to another, enabling the plant to reproduce sexually, mixing genes and producing seeds that will grow into the next generation. This relationship seems simple on the surface, but its intricacy becomes apparent when you look closely. Different flowers have evolved structures specifically adapted to particular pollinators. Long, tubular flowers are perfect for the long tongues of butterflies, or the extendable proboscises of moths. Bowl-shaped flowers accommodate bees that land and walk around while feeding. Some flowers are designed for hummingbirds, with no landing platform at all, because hummingbirds hover while feeding. The colors of flowers are tuned to the visual systems of their pollinators. Bees, for instance, can see ultraviolet light, which humans cannot. Many flowers that appear plain yellow or white to us have striking ultraviolet patterns that guide bees to the nectar, like runway lights directing an aircraft. The timing of flowering is also coordinated with pollinator activity. Spring flowers emerge when bees are beginning to forage after winter. Night-blooming flowers open when nocturnal pollinators like moths or bats are active. In tropical regions, where some plants flower year-round, specific pollinators may depend on a reliable supply of particular flowers, and the plants depend on those pollinators being present. Break this timing through climate change or habitat disruption, and both plant and pollinator suffer. But plant-pollinator relationships are just one category of mutualism. In the oceans, another famous partnership plays out on coral reefs. The clownfish and sea anemone relationship has become iconic, popularized by movies, but the reality is even more interesting than fiction. Sea anemones are animals, not plants, despite their flower-like appearance. They're relatives of jellyfish, and like jellyfish, they're armed with stinging cells called nematocysts on their tentacles. These cells inject toxins that paralyze small fish and invertebrates, which the anemone then pulls to its mouth and consumes. Most fish avoid anemones for obvious reasons, but clownfish have evolved immunity to anemone stings, likely through a combination of a protective mucus coating and physiological resistance to the toxins. A clownfish lives among the anemone's tentacles, darting in and out, completely safe from predators that dare not follow into that forest of stinging arms. The anemone, in return, benefits in multiple ways. The clownfish's movements help circulate water around the anemone, improving oxygen exchange and waste removal. The fish's excrement provides nutrients, particularly nitrogen compounds that the anemone can use. Clownfish also actively defend their anemone home, chasing away butterfly fish and other species that might nibble on the anemone's tentacles. Some researchers even suggest that the bright colors of clownfish may attract potential prey toward the anemone, like a living lure. This relationship is so important that clownfish have become entirely dependent on anemones for protection, particularly when they're young and vulnerable. Without an anemone, a juvenile clownfish rarely survives long. The anemone while not absolutely dependent on clownfish, grows more successfully and reproduces more prolifically when it hosts fish. It's a partnership shaped over evolutionary time, with each species adapting to better coexist with the other. Mutualistic relationships aren't limited to pairs of animals. Some of the most important involve animals and microorganisms. In the human gut, trillions of bacteria belonging to hundreds of species live in a complex ecosystem. These gut microbes aren't parasites, they're mutualistic partners. They break down complex carbohydrates that human digestive enzymes cannot process, particularly fiber. In doing so, they produce short-chain fatty acids that our intestinal cells use for energy. They synthesize vitamins, including vitamin K and several B vitamins, which we then absorb. They compete with and produce compounds that inhibit harmful bacteria, helping protect us from infections. They even influence our immune system, training it and modulating its responses in ways we're only beginning to understand. In return, we provide these bacteria with a stable, warm environment and a steady supply of food. The bacteria that live in our guts have evolved specifically to thrive there. Many cannot survive outside the mammalian digestive tract. This relationship began before we were even fully human. Our primate ancestors hosted similar microbial communities, and these communities evolved alongside our lineage. When we take antibiotics that kill gut bacteria, we often experience digestive problems because the mutualistic community has been disrupted. Restoring gut health sometimes requires fecal transplants, where microbes from a healthy donor are introduced into a patient's gut, effectively reseeding the mutualistic community. This kind of relationship, where microorganisms live inside larger organisms and provide essential services, is called endosymbiosis. It's remarkably common. Termites harbor protists in their guts that digest cellulose, allowing the termites to live on wood. Ruminant mammals like cows have complex microbial communities in their specialized stomachs that break down grass and other plant matter. Aphids host bacteria that produce essential amino acids the aphids cannot make themselves. In each case, the host provides shelter and nutrients, and the microbes provide metabolic capabilities the host lacks. Some endosymbiotic relationships are so ancient and integrated that the partners have become inseparable. The mitochondria in our cells, the organelles that produce most of our energy, were once free-living bacteria. Billions of years ago, a bacterium capable of aerobic respiration was engulfed by a larger cell, and, instead of being digested, took up residence. Over time, most of its genes were transferred to the host cell's nucleus, and it became a permanent resident, an organelle rather than a separate organism. This process, called symbiogenesis, is one of the most important events in the history of life. Without it, complex multicellular organisms like ourselves might never have evolved. Plants underwent a similar event, incorporating cyanobacteria that became chloroplasts, the organelles that perform photosynthesis. The green in every leaf, every blade of grass, every tree, comes from billions of these captured bacteria, now transformed into specialized cellular machinery. These ancient symbioses literally built the world we know. Even more remarkable are the mycorrhizal relationships between plants and fungi. In forests around the world, the roots of most trees are intimately associated with fungi. The fungal threads, called hyphae, wrap around or even penetrate into root cells, creating an interface where nutrients are exchanged. The fungi extend far beyond the roots, spreading through the soil in vast networks. These hyphal networks have been called the wood wide web because they connect different plants, sometimes even plants of different species, allowing them to share resources and information. Through these fungal connections, a plant can access water and nutrients, particularly phosphorus and nitrogen, from a much larger volume of soil than its roots alone could reach. The fungi are far more efficient at extracting these nutrients from soil particles and organic matter. In return, the plant provides the fungi with sugars, products of photosynthesis that the fungi, unable to photosynthesize themselves, cannot make. As much as 30% of the carbon a tree fixes through photosynthesis may be transferred to its fungal partners. But the mycorrhizal network does more than move nutrients. Research has shown that plants connected by these networks can communicate, in a sense. A tree under attack by insects can release chemical signals into the network that stimulate neighboring trees to produce defensive compounds even before they're attacked. A large, established tree can transfer carbon to younger seedlings growing in deep shade, effectively subsidizing the next generation until they're tall enough to reach more light. If one tree is dying, it may transfer its remaining resources to its neighbors, a final act of generosity made possible by the fungal network. Mycorrhizal fungi are so important that most land plants cannot survive without them. When plants first colonized land hundreds of millions of years ago, they likely did so in partnership with fungi. Early land environments were harsh, with thin soils and limited nutrients. The fungi would have been essential for survival, and the relationship has persisted. Today, you can find mycorrhizae in nearly every ecosystem, from tropical rainforests to Arctic tundra, from grasslands to wetlands. Some mutualistic relationships involve 3 or more partners. Leafcutter ants in Central and South American rainforests maintain elaborate underground gardens where they grow fungus. The ants don't eat the leaves they cut. Their digestive systems cannot process plant cellulose. Instead, they feed the leaves to the fungus, which breaks down the cellulose and produces specialized structures that the ants eat. The ants, in turn, tend the fungal gardens carefully, removing competing molds, adjusting humidity, and fertilizing the fungus with their waste. It's an agricultural system that evolved millions of years before humans developed farming. But there's a third partner in this system. The ants host bacteria, often on their bodies, that produce antibiotics specifically targeting the competitor molds that threaten the fungal gardens. Without these bacteria, the gardens would be overrun by parasites. So, it's a 3-way mutualism: ants, fungus, and bacteria, each dependent on the others, working together to sustain the entire system. These partnerships remind us that survival is not solely about individual strength or cleverness. Often, it's about relationships, about cooperation and interdependence. The ecosystems we see around us are not collections of independent actors, but communities of partners, each species connected to others through countless threads of mutual need and benefit. When we protect one species, we're protecting the web of relationships that sustains it. When we destroy one, we may unravel partnerships that have existed for millions of years. Understanding these mutualisms is essential for understanding life itself, for seeing that cooperation, as much as competition, has shaped the living world. The sleep of survival. As autumn deepens and the days grow short, As temperatures fall and food becomes scarce, many animals face a simple problem with no simple solution: how to survive the winter. Migration is one answer, but it requires the energy and ability to travel vast distances. Remaining active is another, but it demands a constant supply of food that may not exist. The third option, evolved independently by diverse groups of animals, is to sleep. Not the nightly sleep that refreshes, but a profound, prolonged sleep that slows life almost to stillness. This is hibernation, and it's one of the most remarkable physiological feats in the animal kingdom. True hibernation is not simply sleeping through the winter. It's a state of profoundly reduced metabolism, where body temperature drops close to ambient temperature. Heart rate slows to a fraction of its normal pace, and breathing becomes so infrequent that an observer might think the animal dead. The metabolic rate, the speed at which the body burns energy, can drop to as low as 2% of normal. An animal that would burn through its fat reserves in weeks if it remained active can make those same reserves last for months by hibernating. The champion hibernators are small mammals like ground squirrels, which face particularly acute challenges. Being small means having a high surface area to volume ratio, which causes rapid heat loss. Staying warm in winter requires tremendous amounts of energy. But being small also means having limited fat storage. A ground squirrel entering winter with insufficient fat reserves will not survive. Those that do have enough fat can enter hibernation, dropping their body temperature from 37 degrees Celsius, the typical mammalian norm, down to near freezing. An Arctic ground squirrel's body temperature during hibernation can fall below 0 Celsius, down to -3 degrees, a state called supercooling, where the body fluids remain liquid despite being below the normal freezing point. At these temperatures, the squirrel's heart rate drops from several hundred beats per minute to as few as 3 or 4. Minutes may pass between breaths. The brain shows minimal electrical activity. All non-essential physiological processes shut down. The animal is in a state called torpor, a temporary hibernation that can last for days or weeks. But interestingly, hibernators don't stay in torpor continuously throughout the winter. Every 1 to 3 weeks, a hibernating ground squirrel will spontaneously arouse, rapidly warming its body back to normal temperature. During these arousal episodes, which last 12 to 24 hours, the animal is fully active, may eat from food stores if available, urinates, and then returns to torpor. These periodic arousals are energetically expensive, consuming a significant fraction of the animal's total winter energy budget. Why animals do this remains somewhat mysterious. One hypothesis is that it's necessary for immune system maintenance, to allow the body to clear infections or repair accumulated damage that cannot be managed during torpor. Another suggests it's related to sleep itself. Paradoxically, animals in torpor may need to periodically wake up in order to get normal sleep, because the neural activity during torpor is so reduced that it doesn't provide the restorative functions that sleep normally does. The process of entering hibernation is complex and carefully regulated. It's triggered by environmental cues, particularly day length, which affects hormonal systems. As days shorten in autumn, changes in melatonin secretion signal the approach of winter. Combined with dropping temperatures and reduced food availability, these cues prepare the animal's body for hibernation. The animal enters a phase called hyperphagia, where it eats voraciously, gaining as much weight as possible. A ground squirrel may double its body weight, packing on fat that will fuel its winter survival. When hibernation begins, the animal doesn't simply cool down passively. It actively suppresses its metabolism and lowers its temperature set point, the internal thermostat that normally maintains body temperature around 37 degrees. The hypothalamus, a brain region controlling body temperature, recalibrates, essentially deciding that cold is now acceptable, even desirable. Metabolic processes slow, reducing heat production. Blood vessels near the skin dilate, allowing heat to escape. The body temperature falls, but this must be done carefully. Cooling too quickly can cause tissue damage, particularly to the brain and heart. The hibernating animal manages its cooling rate, taking hours to reach its torpor temperature. Once there, it maintains a delicate balance, not freezing, but keeping body temperature just above the freezing point of its tissues. If ambient temperature drops further, the animal can even engage in minimal thermogenesis, producing just enough heat to prevent freezing, a form of regulated hypothermia. The biochemical changes during hibernation are profound. Normally, cells require constant supplies of oxygen and glucose to produce energy. During torpor, with metabolism drastically reduced, energy demand is minimal. But this creates new problems. At low body temperatures, many cellular processes that normally occur rapidly slow to a crawl. Cell membranes, made of lipids, become stiffer and less fluid when cold, potentially disrupting the proteins embedded in them. To counteract this, hibernators adjust the composition of their cell membranes before hibernation, incorporating more unsaturated fats that remain fluid at low temperatures. It's like winterizing a car by switching to lighter oil, anticipating the conditions to come. Another challenge is that during torpor, with minimal circulation, some tissues may experience hypoxia, low oxygen levels. Yet, hibernators tolerate this with minimal damage. Research has shown that their cells upregulate protective proteins and antioxidants that prevent oxidative damage when oxygen levels fluctuate. When they arouse and metabolism ramps back up, there's a surge of reactive oxygen species that could harm tissues. But the hibernator's cells are prepared, armed with molecular defenses that neutralize these threats. The brain, usually the most metabolically demanding organ, undergoes dramatic changes. Neuronal connections, synapses, partially retract during hibernation, possibly to conserve energy. When the animal arouses, these connections rapidly reform. It's a dynamic restructuring, breaking and rebuilding the brain's wiring. That would be pathological in a non-hibernator, but is normal and reversible in hibernators. This has intrigued neuroscientists because it suggests that the brain is far more plastic and resilient than previously thought, with potential implications for understanding and treating neurodegenerative diseases. Not all hibernators are created equal. Bears, often cited as hibernators, actually undergo a different process. Bears do sleep through much of the winter, and their metabolism does decrease, but not to the extent seen in small mammals. A bear's body temperature drops only a few degrees, and while its heart rate slows, the bear can wake quickly if disturbed. This state is sometimes called torpor, or winter dormancy, rather than true hibernation. The distinction matters because bears face different challenges. Being large, they lose heat more slowly and can maintain a higher body temperature with less energy expenditure. They also have much larger fat reserves. A bear entering its den may weigh 600 pounds, with a third of that being fat, enough to sustain it for 6 months without eating, drinking, urinating, or defecating. The fact that bears don't urinate during this time is remarkable in itself. Normally, protein breakdown produces urea, a toxic compound that must be excreted by the kidneys. A bear in winter dormancy continues to break down some protein to maintain essential processes. But instead of accumulating urea in its blood, which would be fatal, the bear recycles the nitrogen. Urea is broken down back into amino acids, which are used to build new proteins. It's a closed-loop system, conserving both water and nitrogen, that researchers are studying for potential applications in treating kidney disease and managing astronauts on long space missions. Hibernation is not limited to mammals. Some reptiles and amphibians undergo similar states called brumation, where metabolism slows and the animal becomes inactive for months. Fish in frozen ponds enter torpor at the bottom, where the water, denser at 4 degrees Celsius than at 0 degrees, remains liquid even as ice forms on the surface. Insects too can enter diapause, a state of suspended development that allows them to survive winter as eggs, larvae, pupae, or even adults, depending on the species. But perhaps the most remarkable hibernators are the hummingbirds, which undergo daily torpor. Hummingbirds have the highest metabolic rates of any vertebrate, with heart rates exceeding 1,000 beats per minute and body temperatures around 40 degrees Celsius during active flight. This furious metabolism is necessary to power their hovering flight and to process the enormous amounts of nectar they must consume. But it comes at a cost. Hummingbirds must eat almost constantly while awake, consuming more than their body weight in nectar each day. At night, when they cannot feed, their energy reserves would be exhausted in hours if they maintained their normal metabolism. So, hummingbirds enter torpor each night. Their body temperature drops to 15 or even 10 degrees Celsius, matching the ambient temperature. Their heart rate falls from 1,000 beats per minute to fewer than 50. They become cold and unresponsive, clinging to their perch, essentially hibernating for 10 or 12 hours. In the morning, as light returns, they arouse. This requires a burst of metabolic activity, rapidly burning fat to generate heat. Within 20 or 30 minutes, a hummingbird goes from a cold, nearly comatose state to full wakefulness, ready to resume its frenetic life. This daily cycle of torpor and arousal allows hummingbirds to survive in environments where food is only available during daylight hours. The mechanisms that enable hibernation, torpor, and similar states are subjects of intense research, not just out of curiosity, but because they hold potential medical applications. Understanding how hibernators avoid brain damage during prolonged periods of low oxygen and blood flow could inform treatments for stroke, where blocked blood vessels deprive brain tissue of oxygen. Learning how bears recycle nitrogen and avoid muscle atrophy during months of inactivity could help develop therapies for bedridden patients or astronauts experiencing muscle loss in zero gravity. Insights into how hibernators manage oxidative stress could contribute to treatments for aging-related diseases. There's even been speculation about inducing hibernation-like states in humans for long-duration space travel. A crew hibernating during a multi-year journey to Mars would require less food, water, air, and living space, dramatically reducing mission costs and complexity. While true hibernation in humans remains science fiction, Research into induced hypothermia and metabolic suppression has already yielded medical benefits. Therapeutic hypothermia, cooling patients after cardiac arrest or traumatic brain injury, can reduce tissue damage and improve outcomes, a direct application of principles learned from studying hibernators. As we watch a ground squirrel emerge from its burrow in spring, thin but alive, having survived months of cold and darkness, we're witnessing a feat of biological engineering that rivals any human technology. The squirrel didn't invent hibernation through conscious thought or planning. Evolution did, through countless generations, each small improvement in metabolic regulation, cold tolerance, and energy management increasing the chances of survival. The result is an animal that can turn down its metabolism like a dimmer switch, sleep through the harshest season, and wake in spring ready to resume life. It's a reminder that survival isn't always about strength or speed. Sometimes it's about knowing when to slow down, when to conserve, when to wait patiently for better times to return. And in that patience, powered by intricate physiology, refined over millions of years, lies one of nature's most elegant survival strategies: the power to rebuild. If you've ever watched a gecko drop its tail to escape a predator, or seen a starfish with one arm shorter than the others slowly regrowing, you've witnessed one of biology's most fascinating abilities: regeneration. The power to replace lost body parts, to regrow what has been damaged or destroyed, seems almost magical. Yet, for many animals, it's routine, a survival strategy as fundamental as finding food or avoiding danger. Understanding regeneration is not just about appreciating nature's wonders. It's about unlocking secrets that might one day allow humans to heal injuries that are currently permanent. The undisputed champions of regeneration are the salamanders, a group of amphibians that includes newts and axolotls. A salamander can lose a leg and grow a new one, complete with bones, muscles, nerves, blood vessels, and skin, all perfectly arranged and fully functional. This isn't a partial regrowth or a malformed stub. It's a complete, anatomically correct limb. And salamanders can do this repeatedly throughout their lives. If you amputate a salamander's leg multiple times, it will regrow it multiple times, apparently without limit. The regeneration process begins almost immediately after injury. Within hours, cells near the wound site begin to migrate, covering the injury with a layer of skin. Beneath this skin cap, something remarkable starts to happen. Cells that were once specialized, committed to being muscle cells, bone cells, or cartilage cells, begin to lose their specialization. They dedifferentiate, reverting to a more stem cell-like state. These dedifferentiated cells accumulate at the injury site, forming a structure called a blastema, a mass of proliferating, unspecialized cells that will give rise to all the tissues of the new limb. The blastema is, in essence, a growth zone, similar to the developing limb bud of an embryo. Cells within it receive signals that tell them what to become. Gradually, they re-specialize, some becoming muscle, others bone, others skin, each type organizing itself into the proper position. This process requires precise coordination. The cells must know not only what to become, but where to become it. They must recreate the three-dimensional structure of a limb, with all its complexity, from a formless mass of cells. How they do this is still not fully understood, but it involves a complex interplay of signaling molecules, gene expression patterns, and mechanical forces. One critical element is the nerve supply. Research has shown that regeneration cannot proceed without nerves. If you sever the nerves to a salamander's limb and then amputate the limb, regeneration fails or produces only a small, malformed structure. The nerves supply signals, likely growth factors and other molecules that are essential for blastema formation and growth. Interestingly, the amount of nerve tissue matters. Larger salamanders, with more nerve fibers innervating their limbs, grow larger regenerated limbs. Smaller individuals, with fewer nerves, grow proportionately smaller limbs. Scientists have tested this by grafting blastemas from small salamanders onto large ones. The result? The blastema, receiving signals from the abundant nerves of the large host, grows into a full-sized limb, demonstrating that nerve supply, not just the blastema itself, determines regeneration scale. Salamanders can regenerate more than limbs. They can regrow tails, jaws, portions of the heart, parts of the eye, even sections of the spinal cord. The axolotl, a species of salamander native to Mexico, remains in a juvenile form throughout its life, retaining gills and an aquatic lifestyle. This permanent juvenility, called neoteny, may contribute to the axolotl's exceptional regenerative abilities. Axolotls are extensively used in regeneration research because they're easy to maintain in laboratories, and their regeneration is robust and reliable. Recent molecular studies have begun to uncover the genetic and cellular mechanisms underlying salamander regeneration. Researchers have found that during regeneration, cells ramp up production of proteins extremely quickly, much faster than in non-regenerating tissues or in non-regenerating animals like mammals. This rapid protein production is enabled by large stores of messenger RNA, the molecules that carry instructions from genes to the protein-making machinery. Salamander cells stockpile these mRNA molecules, essentially keeping a large ready-made supply of blueprints. When regeneration begins, cells can immediately start producing the proteins needed for growth and specialization, giving them a head start. Additionally, salamanders have evolved specific sequences within their mRNA that accelerate translation, the process of making proteins from mRNA. These sequences, not found in mammals, allow salamander cells to produce proteins faster. When researchers block these sequences in salamanders, regeneration is impaired, demonstrating their importance. But salamanders aren't the only animals capable of impressive regeneration. Starfish, or sea stars, can regrow lost arms, and some species can regenerate an entire body from a single arm if part of the central disc is attached. Certain species of sea cucumbers can expel their internal organs when threatened, a defense mechanism called evisceration, and then regrow all those organs over several weeks. Flatworms, particularly planarians, are perhaps the most extreme regenerators. Cut a planarian into pieces, and each piece can regenerate into a complete worm. A planarian cut into 50 pieces can produce 50 new worms. This is possible because planarians have a population of stem cells, called neoblasts, distributed throughout their bodies. These neoblasts are pluripotent, capable of becoming any cell type the worm needs. When the worm is cut, neoblasts migrate to the injury sites and begin rebuilding whatever structures are missing, whether that's a head, a tail, or internal organs. Why then can't mammals regenerate like salamanders or planarians? It's a question that has perplexed scientists for decades. Mammals do have some regenerative capacity. Mice can regenerate the tips of their digits if the amputation occurs at the right location, through the last joint. Humans can regenerate fingertips under certain conditions, particularly in children, though the ability is limited and inconsistent. The human liver can regenerate substantial portions of its mass if damaged, but we cannot regrow limbs or organs the way salamanders can. Several hypotheses have been proposed. One is that mammalian wound healing is too fast and too focused on closing the wound quickly, which may prevent blastema formation. When a mammal loses a limb, the wound is rapidly covered with scar tissue, a fibrous mass of collagen that seals the injury but lacks the organization of normal tissue. Scar tissue is strong but inflexible, and it doesn't provide the environment needed for regeneration. In contrast, salamanders form minimal scar tissue, and the tissue they do form is more organized and doesn't block regeneration. Experiments have shown that reducing inflammation in mammals, which drives scar formation, can enhance regeneration, suggesting that inflammation is part of the problem. Another hypothesis involves the immune system. Mammalian immune responses are complex and vigorous, quickly attacking anything foreign or damaged. This may clear away cellular debris too efficiently, removing signals that would otherwise trigger regeneration. Salamanders have simpler immune systems that respond more slowly, possibly allowing regenerative processes to initiate. Studies have shown that macrophages, immune cells that engulf debris, play a crucial role in salamander regeneration. If macrophages are depleted, regeneration fails. But the type and behavior of macrophages matter. They must support regeneration, not just clean up damage. There's also the matter of cellular specialization. Mammalian cells are more rigidly specialized than salamander cells. A mammalian muscle cell is locked into its identity. It cannot easily revert to a stem-like state. Salamander cells, in contrast, retain more plasticity, more ability to switch identities. This may be related to differences in how genes are regulated, which stretches of DNA are accessible and active. Salamanders may have evolved mechanisms to keep more genetic flexibility, allowing cells to de-differentiate when needed. Finally, there's evolution itself. Mammals may have lost extensive regenerative abilities because they didn't need them, or because other adaptations became more important. If rapid wound healing and immune defense increased survival more than regeneration did, natural selection would favor those traits. Regeneration is energetically expensive and takes time, during which the animal may be vulnerable. For fast-moving, warm-blooded mammals, quickly sealing wounds and fighting infections might have been more advantageous than slowly regrowing lost parts. Despite these limitations, the study of regeneration holds immense promise. Researchers are working to identify the key genes and proteins that enable salamander regeneration, with the hope of activating similar pathways in mammals. If we could induce human cells to de-differentiate, form a blastema, and regenerate lost tissue, the medical implications would be staggering. People who have lost limbs to accidents or disease could regrow them. Spinal cord injuries, currently irreversible, might be repaired. Organs damaged by disease could be regenerated rather than transplanted. We're not there yet, but progress is being made. Scientists have identified signaling pathways, such as those involving proteins called FGFs and BMPs, that are crucial for regeneration. They've created genetically modified mice with enhanced regenerative abilities, showing that mammalian regeneration is not impossible, just difficult. They've developed biomaterials that mimic the environment of a blastema, encouraging cells to proliferate and organize rather than simply scar. And they've studied animals like zebrafish, which can regenerate heart tissue, hoping to translate those findings to humans. The animals that can regenerate remind us that the boundaries we perceive in biology are often more flexible than we think. What seems permanent, a lost limb, a damaged organ, may be reversible under the right conditions. The salamander, the starfish, the planarian, each one is a living demonstration that bodies can rebuild themselves, that the genetic instructions for constructing complex structures persist even in adult tissues, waiting to be activated. Understanding how they do this and learning to replicate it is one of the great challenges of modern biology. And the rewards, the ability to heal injuries once thought permanent, to restore function, to give people back what they've lost, would be among the greatest gifts that science could provide. Living light in the darkness, descend into the ocean, past the sunlit surface layers where photosynthesis fuels abundant life, down through the twilight zone where the last glimmers of daylight fade, and into the midnight zone where no sunlight ever penetrates. Here, in perpetual darkness, you might expect to find only blackness, but you would be wrong. The deep sea glitters with light, bioluminescence, light produced by living organisms themselves. Far from being rare, bioluminescence is the rule in the deep ocean. Studies estimate that 3/4 of all animals living in the water column between the surface and 4,000 meters depth can produce light. In the largest habitat on Earth by volume, light comes not from the sun, but from life itself. Bioluminescence is fundamentally a chemical reaction. At its core are two molecules, luciferin, a light-emitting substrate, and luciferase, an enzyme that catalyzes the reaction. When luciferin reacts with oxygen in the presence of luciferase, it undergoes oxidation, releasing energy. In most chemical reactions, this energy dissipates as heat, but in bioluminescence, the energy is released as light, a process so efficient that virtually no heat is produced. It's called cold light, and it's far more efficient than any light bulb we've invented. Different organisms have evolved different types of luciferin and luciferase, a clear sign that bioluminescence has evolved independently many times. Estimates suggest it has arisen at least 40 times across the tree of life, in bacteria, dinoflagellates, fungi, insects, squid, fish, and many other groups. Each lineage has its own version of the chemistry, tuned to its specific needs. Some produce blue light, which travels farthest through water. Others produce green, red, or even yellow light, colors that are less common but serve specialized purposes. The most common color in the deep sea is blue-green, with a wavelength around 470 nanometers. This is not arbitrary. Water absorbs light, and it absorbs longer wavelengths, like red and yellow, much more strongly than shorter wavelengths like blue. In clear ocean water, blue light can travel 100 meters or more, while red light is absorbed within a few meters of the surface. For deep-sea animals communicating or hunting with light, blue is the logical choice. It travels far, making it effective for signaling or illuminating prey. Some animals, however, have evolved the ability to produce red light, even though red is uncommon in the deep sea. The dragonfish is a notable example. It has light-producing organs beneath its eyes that emit red light, a color most other deep-sea creatures cannot see. By shining red light on potential prey, the dragonfish effectively uses an invisible flashlight, illuminating its target without alerting it or attracting the attention of predators. It's like having night vision goggles in a world where everyone else is colorblind to red. Bioluminescence serves many functions. For some animals, it's a lure. The anglerfish is perhaps the most famous example. Female anglerfish have a modified dorsal spine that extends forward over the head like a fishing rod. At the tip of this rod, called an esker, is a glowing lure, a culture of bioluminescent bacteria that the fish cultivates in a specialized light organ. The bacteria receive nutrients from the fish's bloodstream and, in return, produce a steady glow. Smaller fish, attracted by what appears to be a morsel of food or a cloud of glowing plankton, approach the lure. When they get close enough, the anglerfish strikes, its enormous jaws snapping shut with incredible speed. The anglerfish doesn't hunt actively, it sits motionless in the darkness, letting its lure bring food to it. For other animals, bioluminescence is defense. The vampire squid, despite its ominous name, is a small, passive creature that feeds on detritus drifting through the water. When threatened by a predator, it doesn't flee quickly. It's not a fast swimmer. Instead, it wraps its webbed arms around itself, inverting them so that the spiny undersides face outward, turning itself into a spiky ball. At the same time, it ejects a cloud of glowing mucus from the tips of its arms. The mucus, filled with bioluminescent particles, creates a dazzling, disorienting display. The predator, momentarily confused by the light show, pauses or veers away, and the vampire squid uses that precious moment to escape. Another defensive use is the burglar alarm strategy. The Atolla jellyfish, when attacked, produces rapid, bright flashes of blue light arranged in a spinning pinwheel pattern. This isn't meant to scare the attacker. It's meant to attract a larger predator. The jellyfish essentially says, "There's something here worth attacking," hoping to draw in a predator of its attacker, which might become distracted or frightened and release the jellyfish. It's a desperate tactic, but in the dark ocean it can work. Perhaps the most widespread use of bioluminescence in the deep sea is counter-illumination. Many fish and squid have light organs, called photophores, arranged on their undersides. When viewed from below, these animals would normally be silhouettes against the faint light filtering down from above, making them easy targets for predators hunting from deeper water. But by adjusting the intensity of their photophores to match the downwelling light, they can eliminate their silhouette, blending in with the background. It's active camouflage, constantly adjusted as the animal moves to different depths or as ambient light changes. Lanternfish, small fish that are among the most abundant vertebrates on the planet, use counter-illumination extensively. During the day, they remain in deeper, darker water. At night, they migrate upward to feed in shallower, more productive waters, and their bioluminescence helps them avoid predators during this daily journey. Not all bioluminescence comes from the animals themselves. Many species host symbiotic bacteria that produce light. The Hawaiian bobtail squid has a specialized light organ in its mantle cavity, colonized by a specific species of bioluminescent bacteria shortly after the squid hatches. The squid carefully regulates this bacterial population, culling it daily and allowing it to regrow, maintaining a controlled culture. The bacteria glow constantly, but the squid can modulate and direct the light using muscles, lenses, and reflectors in the light organ, essentially using the bacteria as a biological flashlight. In return, the bacteria receive a safe, nutrient-rich environment. It's a mutualistic relationship, squid and bacteria evolving together. The evolutionary origins of bioluminescence are still being studied, but one hypothesis suggests it may have first arisen as a way to deal with oxygen, which, while essential for respiration, also creates toxic reactive compounds. The luciferin-luciferase reaction consumes oxygen, and in doing so, it may have helped early organisms manage oxygen toxicity. The light was initially a byproduct, but once it existed, natural selection could co-opt it for communication, predation, and defense. Over time, organisms refined the chemistry, developed specialized light organs, and integrated bioluminescence into their ecology. The diversity of bioluminescent organs is astounding. Some are simple clusters of light-producing cells. Others are complex structures with lenses to focus the light, reflectors to direct it, color filters to change its hue, and shutters to control when light is emitted. The photophores of some deep-sea fish are so sophisticated that they resemble miniature eyes, except instead of detecting light, they produce it. This convergence, the independent evolution of similar structures for different purposes, highlights how strongly natural selection shapes organisms. Bioluminescence research has practical applications beyond simple curiosity. The genes for luciferase have been invaluable in molecular biology. Scientists use them as reporter genes, tags that make cells glow when a particular gene is active. This allows researchers to visualize gene expression in real time, to watch as cells respond to signals or undergo developmental changes. Luciferase from fireflies is commonly used in laboratories around the world, helping advance research in genetics, cancer biology, neuroscience, and many other fields. There's also potential in using bioluminescent bacteria for bioremediation, cleaning up pollution. Some bacteria glow in the presence of specific pollutants, providing a living sensor system. Bioluminescence could even be harnessed for sustainable lighting, though current technologies are far from practical. Imagine trees that glow at night, providing street lighting without electricity. It's speculative, but it's rooted in real biology. As we explore the deep sea with submersibles and remotely operated vehicles, we continue to discover new bioluminescent species, new variations on the theme of living light. Each one adds to our understanding of how life adapts to extreme environments, how evolution solves the problem of communication and survival in a place where sunlight never reaches. The deep ocean, once thought to be a barren, lifeless void, is in fact a bustling ecosystem, lit not by the sun, but by the collective glow of billions of organisms, each one a tiny beacon in the darkness. It's a reminder that even in the most extreme and inhospitable places, life not only survives but flourishes, finding ways to thrive that we are only beginning to understand. The great journeys. Every autumn, as days shorten and temperatures drop, something remarkable begins. And just backyards and forests across eastern North America, monarch butterflies, insects weighing barely half a gram, launch themselves into the air and begin flying south. They won't stop until they reach the Oyamel fir forests of central Mexico, a journey of up to 3,000 miles. No individual butterfly has made this trip before. The butterflies migrating south are several generations removed from those that made the journey in the opposite direction, the previous spring. Yet somehow, with no guide, no prior experience, they navigate with precision to specific mountain groves, often to the very same trees where their ancestors spent the previous winter. Migration, the large-scale, directed movement of animals from one region to another, is one of the most spectacular phenomena in nature. It occurs in nearly every major animal group: insects, fish, birds, mammals, even some crustaceans and reptiles. The journeys vary in scale from a few miles to distances that span hemispheres. But all migrations share common features. They are predictable, often seasonal, and they're driven by the need to access resources, food, breeding sites, or favorable climates that vary in time and space. The navigation challenges posed by migration are immense. To successfully migrate, an animal must know where it is, where it needs to go, and how to get there. This requires not just a compass, a sense of direction, but also a map, a sense of position. And it requires the ability to integrate multiple sources of information, compensating for winds, currents, and other factors that can push an animal off course. Many migratory animals use the sun as a compass. The sun's position in the sky changes predictably throughout the day, rising in the east, reaching its highest point at noon in the southern sky, in the northern hemisphere, and setting in the west. An animal with an internal biological clock can compensate for the sun's movement, determining a fixed compass direction. Starlings, pigeons, and many other birds have been shown to use sun compasses. Even bees use the sun's position to communicate the location of food sources through their waggle dance, encoding both distance and direction relative to the sun. But what about cloudy days when the sun isn't visible? Many insects and birds can detect the polarization pattern of skylight. Sunlight becomes polarized as it scatters through the atmosphere, creating a pattern across the sky that's invisible to us but detectable by animals with the right visual adaptations. This polarization pattern is consistent and reliable, even when clouds obscure the sun itself. Bees, ants, and many migratory insects use polarized light for navigation. Some birds do as well, though they also rely on other cues. At night, when the sun is down, nocturnal migrants, like many songbirds, use stars. Studies with caged migratory birds in planetariums have shown that birds can orient themselves by the star pattern. Interestingly, they don't seem to use specific stars as references, like sailors using Polaris, the North Star. Instead, they use the overall pattern of stars, particularly the rotation of the night sky around the celestial pole. Young birds must learn this pattern. It's not entirely innate. Experiments where birds are raised under artificial skies with shifted star patterns show that the birds later orient according to the artificial sky they learned, not the real one. Perhaps the most fascinating and navigational tool is the Earth's magnetic field. Our planet acts like a giant magnet, with magnetic field lines running from pole to pole. These field lines have two properties that can provide navigational information: their inclination, the angle at which they intersect the Earth's surface, and their intensity, which varies with latitude. Near the equator, field lines are nearly horizontal, and intensity is low. Near the poles, they're nearly vertical, and intensity is high. An animal that can detect these properties has access to a latitude map. Many animals possess a magnetic sense. Sea turtles, salmon, birds, bats, and even fruit flies have been shown to respond to magnetic fields. The mechanisms underlying this sense are still being investigated, but two main hypotheses have emerged. One involves magnetite, a naturally occurring magnetic mineral. Some animals have tiny crystals of magnetite in their tissues, particularly in the beak or head region in birds. These crystals, coupled to sensory nerves, could act as a compass, physically pulling or pushing in response to the magnetic field and triggering nerve signals. The second hypothesis involves a quantum mechanical process in the eye. Certain proteins called cryptochromes, found in the retina, are sensitive to magnetic fields due to the behavior of electron pairs. When light hits these proteins, it can create pairs of electrons with their spins entangled, a quantum state. The Earth's magnetic field affects these spin states, altering the chemical reactions that follow. This produces signals that the bird's nervous system can interpret as magnetic direction. It sounds like science fiction, but experimental evidence supports it. Birds lose their magnetic sense when specific wavelengths of light are blocked, consistent with the cryptochrome hypothesis. Magnetic sense provides a compass, but what about a map? How does a bird, displaced hundreds of miles from its usual route, find its way back? This is the map part of navigation, determining one's position on the Earth's surface. Some animals appear to create cognitive maps, mental representations of landmarks and spatial relationships. Pigeons, released far from their home loft, will circle to gain altitude, presumably surveying the landscape for familiar features before heading home. If transported in dark containers that prevent them from seeing landmarks en route, they're disoriented when released, suggesting they rely on landscape memory. But other animals migrate over vast, featureless expanses, oceans or open skies, with no landmarks to guide them. Salmon, for instance, spend years in the open ocean before returning to spawn in the exact river, even the specific stream where they were born. How do they do this? Olfaction, the sense of smell, plays a critical role. Young salmon imprint on the chemical signature of their natal stream, a unique combination of dissolved minerals, plant compounds, and other substances. When they mature and return from the ocean, they navigate using this olfactory memory, swimming upstream, testing the water, and following the scent gradient that leads home. Some evidence suggests that salmon also use magnetic cues, imprinting on the magnetic signature of their birthplace. The Earth's magnetic field varies slightly from place to place due to local geology. By remembering the magnetic field characteristics of their natal stream, salmon may supplement their olfactory map with a magnetic one, providing a backup navigation system. Birds migrating across oceans face similar challenges. The Arctic tern holds the record for the longest migration of any animal, flying from the Arctic to the Antarctic and back each year, a round trip of over 40,000 miles. These terns see more daylight than any other animal, experiencing summer at both poles. Their migration is multimodal. They use the sun, stars, magnetic fields, and likely visual landmarks when available. But much of their journey is over open ocean, featureless water stretching to the horizon. Here, subtle cues, the angle of waves, the patterns of wind, even possibly infrasound, very low-frequency sounds from distant storms or ocean waves, may provide information. The genetic and hormonal regulation of migration is complex. Many migrants exhibit restlessness, called Sugenruhe, before and during the migration season. Caged migratory birds will hop and flutter at the times they would normally be migrating, oriented in the direction of migration. This restlessness is driven by changes in hormone levels, particularly prolactin, thyroid hormones, and corticosterone, which are themselves responsive to environmental cues like day length and temperature. The migratory urge is partly hardwired, controlled by genetic programs that specify when to migrate and in which direction. But it's also flexible, adjustable based on conditions and experience. Young birds often migrate with adults, learning the route. But in some species, like cuckoos, which lay eggs in other birds' nests, the young never meet their parents. Yet these young cuckoos, raised by host species, still migrate to the correct wintering grounds at the correct time, demonstrating that the basic migratory program, timing, direction, and distance, is innate, encoded in their genes. Migration is energetically expensive and dangerous. Birds must accumulate enough fat to fuel journeys that may take days or weeks. They face predators, storms, starvation if they miscalculate. Many die en oxysexed route. Yet the benefits, access to abundant food resources, favorable breeding conditions, escape from harsh winters, outweigh the costs. Migration shapes ecosystems, transporting nutrients, genes, and individuals across landscapes, connecting distant habitats. Human activities are disrupting migrations. Habitat destruction removes critical stopover sites where migrants rest and refuel. Artificial light disorients nocturnal migrants, causing collisions with buildings. Climate change shifts the timing of food availability, potentially creating mismatches where migrants arrive too early or too late. Understanding migration is not just academic, it's essential for conservation. As we watch a flock of geese flying in V formation overhead, or stand on a beach as sea turtles emerge from the waves to nest, we're witnessing journeys that have occurred for millions of years. These animals are following paths laid down by their ancestors, guided by senses we barely understand, navigating challenges that would daunt us even with all our technology. Migration is a testament to the power of instinct and adaptation, a reminder that life is not static but dynamic, flowing across the planet, following cycles older than humanity itself. Surviving the extremes. Life, at its core, is a delicate balance. Temperatures too high or too low disrupt proteins, freezing or denaturing them. Pressures too great can crush cells. Too much or too little water, oxygen or salt can be equally fatal. Most organisms can only survive within narrow ranges of these conditions, but evolution has repeatedly found ways to push the boundaries, creating species that thrive in environments that would be instantly lethal to most life. These extremophiles and their adaptations show us just how versatile and resilient life can be. Deserts are perhaps the most visibly harsh terrestrial environments, places where water is scarce. Temperatures soar during the day and plummet at night, and plants are few and far between. Surviving here requires solving the problem of water conservation. The kangaroo rat, a small rodent living in the deserts of North America, has become a master at this. It can live its entire life without ever drinking liquid water. All the moisture it needs comes from the seeds it eats. This is possible because of extraordinary physiological adaptations. The kangaroo rat's kidneys are phenomenally efficient, producing urine so concentrated that it's nearly solid. While human urine is about 2% urea by weight, kangaroo rat urine can exceed 25%. This extreme concentration minimizes water loss. The kangaroo rat also recovers moisture from exhaled air. As it breathes out, warm, moist air from the lungs passes through the nasal passages. These passages are cooler, and moisture condenses on them. When the rat inhales, it reabsorbs some of this water. It's a recycling system, reclaiming water that would otherwise be lost. Additionally, kangaroo rats are nocturnal, remaining in cool, humid burrows during the scorching day. They seal the burrow entrance with soil, trapping moisture and creating a microclimate that's far less harsh than the surface. Their behavior, physiology, and anatomy all work together to conserve water. Other desert animals have different strategies. The fennec fox, the smallest fox species, has enormous ears, disproportionately large for its body. These ears are not just for hearing, they're radiators. The dense network of blood vessels in the ears dissipates heat, allowing the fox to cool itself without sweating, which would waste water. Many desert reptiles avoid the heat by being active at dawn and dusk, the cooler twilight hours. Some estivate, entering a state of dormancy during the hottest, driest part of the year, much like hibernation, but triggered by heat and aridity rather than cold. At the opposite extreme, animals in polar regions face cold so intense that exposed skin can freeze in minutes. Survival here requires insulation and the ability to maintain body temperature. Penguins living in Antarctica have dense feather layers that trap air, providing exceptional insulation. Their feathers are so tightly packed that water cannot penetrate, keeping skin dry. Beneath the feathers, a thick layer of blubber provides additional insulation and energy storage. Penguins huddle together during the worst storms, reducing heat loss by sharing warmth. The individuals on the outside of the huddle gradually rotate to the inside, ensuring that no penguin remains exposed for too long. It's a social adaptation, collective thermoregulation. Polar bears, the apex predators of the Arctic, are similarly insulated. Their fur, which appears white but is actually translucent, scatters light, providing camouflage. Beneath the fur, black skin absorbs solar radiation, converting it to heat. A thick layer of blubber, up to 4 inches thick, provides both insulation and energy. Polar bears are so well insulated that they can overheat if they run too much. Their primary concern isn't staying warm, it's avoiding overheating while hunting, a problem few other Arctic animals face. In the oceans, life extends to the deepest trenches, places where pressure exceeds 1,000 atmospheres, enough to crush most submarines. At these depths, there is no light, temperatures hover just above freezing, and the pressure is immense. Yet fish, shrimp, and other organisms live here. How do they withstand such pressure? The key is that they have no air spaces. Air compresses dramatically under pressure, which is why divers risk lung and ear injuries if they ascend too quickly. But water is virtually incompressible. A deep-sea fish's body is mostly water, with cells, tissues, and fluids all at the same pressure as the surrounding environment. There's no pressure gradient pushing inward, so the fish isn't crushed. However, pressure does affect biochemistry. Proteins, which depend on specific three-dimensional shapes to function, can be distorted by high pressure. Deep-sea organisms produce versions of key proteins that are pressure adapted, maintaining their functional shapes even under extreme conditions. Some deep-sea fish have replaced certain molecules in their cells with ones that stabilize proteins under pressure. For instance, they accumulate trimethylamine oxide, a small organic molecule that counteracts the destabilizing effects of pressure on proteins. It's molecular adaptation at the biochemical level. Perhaps the most extreme environments on Earth are hydrothermal vents, undersea volcanic springs, where superheated, mineral-rich water erupts from the seafloor. The water temperature can exceed 400 degrees Celsius, hot enough to melt lead, though the extreme pressure keeps it liquid. The water is also laden with toxic chemicals, hydrogen sulfide, which would be lethal to most life, heavy metals, and acidic compounds. Yet vents are oases of life. Giant tube worms, over 2 meters long, cluster around vents, their bright red plumes waving in the current. These worms have no mouth, no digestive system. They cannot eat. Instead, they host chemosynthetic bacteria in a specialized organ called a trophosome. These bacteria use the hydrogen sulfide from the vent water as an energy source, combining it with oxygen from seawater to produce energy, much like we use oxygen and glucose. In the process, the bacteria produce organic molecules, sugars, and amino acids, which the worm absorbs. It's a symbiosis as intimate as any on Earth, with the worm entirely dependent on its bacterial partners. Other vent animals have similar partnerships, or feed on the bacteria and the creatures that eat them, creating a food web independent of sunlight. Photosynthesis, the basis of nearly all surface ecosystems, is irrelevant here. Energy comes from chemistry, not from the sun, a reminder that life is versatile, finding pathways wherever energy is available. High-altitude environments present their own challenges, primarily low oxygen levels. At the summit of Mount Everest, the atmospheric pressure is only 1/3 of what it is at sea level, meaning each breath delivers far less oxygen. Humans suffer acute mountain sickness, confusion, difficulty breathing, even death if exposed without acclimatization. Yet some animals live comfortably at these altitudes. The bar-headed goose migrates over the Himalayas, flying at altitudes exceeding 7,000 meters, higher than most commercial jets cruise. To do this, it has evolved hemoglobin with a higher affinity for oxygen, allowing it to extract more oxygen from thin air. Its cardiovascular system is also adapted, with a larger heart and more efficient lungs. Vicuñas, relatives of llamas, live year-round in the Andes at altitudes above 4,000 meters. Their blood contains more red blood cells and hemoglobin than lowland animals, increasing oxygen-carrying capacity. Their hearts are larger relative to body size, pumping more blood with each beat. Even their cells are adapted, with mitochondria that function more efficiently under low oxygen conditions. These adaptations are partly genetic, evolved over thousands of generations, and partly developmental, the result of growing up at high altitude. These examples, from deserts to polar ice, from the deepest ocean trenches to the highest mountains, from scorching vents to frozen tundra, illustrate a profound truth. Life is not constrained to a narrow, comfortable zone. Evolution has repeatedly found solutions to the most extreme challenges, crafting species that thrive where others cannot even survive. Each adaptation, whether behavioral, physiological, or biochemical, represents a solution to a specific problem, tested through the unforgiving filter of natural selection. Studying extremophiles has practical implications. Enzymes from thermophilic bacteria that live in hot springs are used in PCR, a technique that amplifies DNA and is fundamental to modern genetics. Antifreeze proteins from polar fish are being explored for use in cryopreservation and even in ice cream to prevent crystallization. Understanding how deep-sea organisms manage pressure could inform the design of submarines and deep-sea equipment. Each adaptation, each molecular trick, is a potential tool for human use. But beyond applications, extremophiles teach us humility. They remind us that life is far tougher, far more creative than we often assume. The planet is not just the surface environments we see daily, it's also the crushing depths, the searing vents, the frozen wastes, and the parched deserts. And in each of these places, evolution has found a way. The animals that live in these extremes are not oddities or exceptions. They're demonstrations of life's fundamental capacity to adapt, to survive, to thrive against all odds. They show us that survival is not about living in comfort. It's about finding a way, any way, to persist, to reproduce, to pass on genes to the next generation. And in that persistence, in that stubborn refusal to yield, lies the essence of life itself. Closing thoughts. As we come to the end of tonight's journey through nature's greatest survival strategies, take a moment to reflect on what we've discovered. We've seen tardigrades pause their lives, entering suspended animation that allows them to survive the vacuum of space. We've watched cephalopods dissolve into their surroundings, masters of disguise changing colour and texture in fractions of a second. We've explored the intimate partnerships between species, mutualisms where neither partner can survive alone, from clownfish sheltering among anemone tentacles to vast fungal networks connecting forest trees. We've witnessed the profound metabolic suppression of hibernation, where a ground squirrel's heartbeat slows to a crawl and its body temperature drops to near freezing, only to revive months later as if no time had passed. We've marveled at salamanders regrowing complete limbs and planarians regenerating entire bodies from tiny fragments, demonstrations that the boundaries of biological possibility are far more flexible than we imagine. We've descended into the dark ocean where 3/4 of all animals produce their own light, turning chemistry into cold luminescence that illuminates the abyss. We've followed migrations spanning continents and oceans, animals navigating by sun, stars, magnetic fields, and senses we're only beginning to understand. And we've visited the extremes of Earth, deserts and deep seas, polar ice and volcanic vents, finding life thriving in places that should be barren. Each strategy we've explored tonight is the product of millions of years of refinement. Each one is a solution to a problem that evolution posed and that life answered. But these strategies are not just historical curiosities. They're living blueprints, demonstrations of what's possible when biology is pushed to its limits. They hold lessons for medicine, engineering, conservation, and our understanding of life itself. As you drift towards sleep tonight, perhaps you'll dream of tiny water bears curled into tunes, waiting patiently in suspended animation for water to return. Or of cuttlefish flickering through colour patterns, conversations in light. Or of salmon returning home to streams they left years before, guided by memory and instinct. Or of lights glowing in the deep ocean, conversations and hunts carried out in bioluminescent flashes. Remember that you too are part of this ancient story. The air you breathe is produced by photosynthetic organisms that conquered land in partnership with fungi. The bacteria in your gut help you digest food in a mutualism as important as any coral and algae. Your cells carry mitochondria that were once free-living bacteria, a symbiosis billions of years old. You are connected through evolution and ecology to every organism we've discussed tonight. If you enjoyed this exploration of nature's survival strategies, please like this video and subscribe to Bedtime Biology. Share your thoughts in the comments, tell us where you're watching from, and let us know what topics you'd like us to explore next. Sweet dreams, and may your rest be as peaceful as a hibernating bear, as resilient as a tardigrade, and as wonder-filled as a night ocean lit by living light.